Chapter 10 of our behavioral neuroscience textbook introduces one of the major and probably more complicated sensory systems, which is vision. So vision serves basic needs in an evolutionary standpoint. We developed a certain vision to find food, avoid predators, find a mate, and locate shelter. And about 1/3 of the cerebral cortex is devoted to visual analysis and perception. So to say vision is important would be an understatement. So the visual system extends from the eye to the brain. It follows the path of information triggered by light, starting with the optical properties of the eye. And then we'll look at the cells and processes intrinsic to the back of the eye that encode light as neural activity. And thirdly, the information flowing from the eye to the cortex. So in order to start to introduce the different structures of the eye, the textbook compares the eye to a camera, which the reason why will reveal itself in very shortly. So the cornea, which is the outer layer of the eye and whose curvature is fixed, bends light rays and is primarily responsible for forming the image on the retina. The lens, which is the structure in the eye that helps to focus an image on the retina. The light rays travel straight until they hit the cornea, and it is there where they bend, which is called refraction. And unlike in fishes, amphibians, and reptiles, whose lenses move in and out to adjust focus like a camera, the birds and mammals' focus is adjusted and controlled by ciliary muscles, which change the shape of the lens, and this process is called accommodation. And so the reason why we wear glasses when we're old is because as we age, our lenses become less elastic and therefore less able to change curvature to bring nearby objects into focus. And so the amount of light that enters the eye is controlled by our pupils, which are the holes in our eyes, the black holes, and it is formed by the iris, which is the colorful ring surrounding it. And the pupil can be dilated or constricted, and the dilation is controlled by the sympathetic nervous system, and when it's constricted, it's being controlled by the parasympathetic nervous system. So drugs can be used to induce this dilation and constriction. So, for example, belladonna and similar drugs may be used to block acetylcholine transmission in the parasympathetic synapses of the iris in order to keep it open. Meanwhile, other drugs like morphine constrict the pupil. And so the movement of the eyes is controlled by extraocular muscles, which are 3 pairs of muscles that extend from the outside of the eyeball to the bony eye socket that we call the orbit. So the previously mentioned eye structure, the retina, is where visual processing takes place. So the retina is the receptive surface inside the back of the eye that contains photoreceptors and other neurons. And as I said, visual info processing occurs here. And so the receptor cells in the retina that detect light are called photoreceptors, and there are 2 major kinds: rods, which are most active at low levels of light, and cones, which are responsible for color vision. And both of these photoreceptors release neurotransmitter molecules that control the activity of the bipolar cells, which are interneurons that receive info from rods and cones and pass the information to retinal ganglion cells, whose axons form the optic nerve, which is the collection of ganglion cell axons that extends from the retina to the optic chiasm and carries information from the eyes to the brain. And there are also specialized retinal cells two kinds. The first kind is called horizontal cells, which contact both receptor cells and bipolar cells, and demacron cells, which contact both bipolar cells and ganglion cells and are especially significant in inhibitory interactions within the retina. And so there are two different systems as well. The scotopic system, which has only one receptor type, rods, and thus does not respond to different colors, and the photopic system, which uses cones and requires more light, and in some species shows differential sensitivity to wavelengths and thus enables heightened color vision. So the photoreceptors transduce light into chemical reactions. So only a fraction of the light that passes through the cornea actually reaches the retina, and the stacking disc structure of rods and cones increase the likelihood that one will capture those light particles, called photons. And the photons that strike the discs are captured by special photopigment receptor molecules, and rods, they are called rhodopsin. And these photopigments consist of two major parts, opsin and retinal. So to further explain light and how that works, box 10.1, which is on page 304, talks about electromagnetic radiation, which is the physical energy to which our visual systems respond to. This energy comes in small packets called quanta, or quantum as the plural, and each packet can be described by a number, its wavelength, which is the distance between two adjacent crests of vibrational activity. And photons are the special quanta that fall in the range of 400 to 700 nm, and these are the quanta that the human visual system responds to. So as previously mentioned, the visual system can adjust the size of the pupil. This is one mechanism that enables the eyes to work over a wide range of light intensities. So a process called range fractionation, which is the handling of different intensities by different receptors. Rods have low thresholds and cones have high thresholds, is one other mechanism. And natural selection gave each photoreceptor an enormous range of adaptation, meaning they can adjust its sensitivity to match the ambient illumination. And photoreceptor adaptation accounts for us being able to see over such wide ranges of light. And there are two main factors for most of adaptation, and those are varying concentrations of calcium ions and the slow recombining of retinal and opsin when the photoreceptor is split apart by light. So the last section of the segment talks about how acuity is best in the foveal vision. And so our visual field is the whole area that we can see without moving our heads or eyes. And visual acuity is the sharpness of vision, which is especially fine in the center of the field and a little less so towards the periphery. And within the retina, the fovea is the central region that has a dense concentration of cones. And the optic disc is the nasal side of the fovea where blood vessels and ganglion cell axons leave the eye. So it is in the optic disc that we have a blind spot, because in this part there aren't any photoreceptors. So visual acuity is largely determined by high concentrations of cones. So different people have high levels while others have low levels. And some animals have higher levels than others as well. For example, hawks have a much sharper vision than we do because they have a higher concentration. So another important term to know is saccades, which are quick movements of the eye, which bring various parts of the visual scene to the fovea to take advantage of the sharp visual acuity. And so when we fix our gaze, small saccades that we are even unaware of avert photoreceptor adaptations. And the last point to take into account is that rods are absent in the fovea but more numerous than cones in the periphery, and the rods are more sensitive to dim light than cones, and so provide a high sensitivity with limited acuity, while cones provide high acuity with limited sensitivity.